The story started when three friends are having a lunch together.
Oh, I'm so hungry.
Firstly, I will be explaining about carbohydrates. So this will briefly explain about what happens in our body when we consume from carbohydrate sources or when it gets depleted. When we have eaten, our blood glucose level will increase, producing secretion of glucagon by the pancreas to convert glycogen to glucose. And when we are having depletion from the source carbohydrates, pancreas will secrete insulin to convert glucose to glycogen in order to maintain human's body glucose level. There are some key processes in our body that aid in homeostasis. For example, when our sugar levels rise up, glycogenesis, which is the glycogen synthesis from glucose, will occur. And when sugar levels drop, ketogenesis, which is the production of ketone bodies, gluconeogenesis, which is synthesis of glucose from non-carbohydrate precursors, and glycogenolysis, which is the breakdown of glycogen to glucose, will occur. For this video, I will be focusing more on gluconeogenesis and glycogenolysis. As I have mentioned before, the process glycogenolysis is a breakdown of glycogen to glucose. As we can see in the diagram, firstly, the glycogen will be converting into glucose 1-phosphate by the addition of inorganic phosphate and the help of glycogen phosphorylase and the branching enzyme. Next, it will be converting into glucose 6-phosphate by the help of phosphoglucomutase and magnesium ion. And lastly, it will produce glucose by the help of glucose 6-phosphatase. Meanwhile, gluconeogenesis is the production of glucose by non-carbohydrate precursor. As we see in the diagram, I've stated three examples, which is amino acid, glycerol, and lactate. So from pyruvate, it will convert into oxaloacetate by the help of pyruvate carboxylase, continuing on phosphoranyl pyruvate by the help of PEP carboxy kinase, converting into fructose 1,6-biphosphate, and then fructose 6-phosphate by the help of fructose 1,6-biphosphate, converting into glucose 6-phosphate, and at last becoming glucose by the help of glucokinase.
How about lipid metabolism during starvation? During starvation, blood glucose level in our body is low. In response to low blood glucose level, the secretion of insulin from the pancreatic cells also decrease. However, another hormone called as glucagon is released. Glucagon triggers lipolysis and indirectly reducing lipogenesis. Lipolysis process releases a product of fatty acid and glycerol. This fatty acid requires an activation to form fatty acyl-CoA in order to undergo beta oxidation, forming acetyl-CoA. Then, ATP can be produced through citric acid cycle. The excess of acetyl-CoA produced will be converted into ketone bodies through a process called as ketogenesis. Ketone bodies such as beta-hydroxybutyrate, acetoacetate, and acetone serve as an alternative energy source, particularly for the brain and other tissue. As I mentioned earlier, lipolysis process is triggered during starvation. So what happened during lipolysis process? Stored fats in adipose tissue are mobilized to provide fatty acids for energy production. Lipolysis involves the process of breaking down a triglyceride into a glycerol and 3 fatty acids through hydrolysis reaction. The enzymes involved in this whole process of lipolysis are adipose triglyceride lipase, hormone-sensitive lipase, and monoglyceride lipase. There are also 3 hormones involved in lipolysis, which is adrenaline, cortisol, and glucagon.
The general idea of beta oxidation. First oxidation, removal of 2 hydrogen from a fatty acyl-CoA, formation of double bond. Second is hydration, addition of water across the double bond, formation of hydroxyl group. The third one is dehydrogenation. Another set of hydrogen atoms are removed from the beta carbons, a formation of a trans double bond. Lastly is the thiolysis. Enzyme thiolase cleave the beta ketoacyl-CoA into acetyl-CoA, then shorten the fatty acyl-CoA chain. Beta oxidation. Fatty acid in the formation of an alkene in an oxidation reaction catalyzed by the acyl-CoA dehydrogenase. This reaction, the coenzyme FAD accepts two hydrogen atoms from the acyl-CoA from the alpha carbon and one from the beta carbon, forming the reduced flavin adenine dinucleotide FADH2. Next, the trans-alkene is hydrate form a secondary alcohol in reaction catalyzed by enol-CoA hydratase. Enzyme form only the L isomer. Secondary alcohol is then oxidized to a ketone by the beta-hydroxyl acyl-CoA dehydrogenase with NADA+ acting as the oxidizing agent. The oxidation of each molecule of NADH to NAD+ by electron transport chain will furnish 2.5 to 3 molecule of ATP. Final reaction is cleavage of beta-ketoacyl-CoA, a molecule of coenzyme A. The products are acetyl-CoA and fatty acyl-CoA have been shortened by 2 carbon atoms. Reaction is catalyzed by the thiolase. Shortened fatty acyl-CoA is then degraded. Repetitions of these 4 steps time will result a molecule of acetyl-CoA. Ketogenesis, period of high beta oxidation or when the citric acid cycle is unable to fully process the acetyl-CoA generated, excess acetyl-CoA accumulates. Excess acetyl-CoA is diverted to ketogenesis where it undergoes condensation reaction to form ketone bodies, primarily acetoacetate. Ketone bodies being water-soluble can easily enter the bloodstream. They are released from liver into the circulation. Ketone bodies circulate in the bloodstream and can be uptaken by the extravascular tissue such as the brain, heart, and skeletal muscle. In peripheral tissue, ketone bodies are converted back into acetyl-CoA, which enters the citric acid cycle for energy production.
Next, we go into protein. The protein is the last source of our body during starvation, after the carbohydrate and lipid are depleted. Protein will undergo photolysis to break down in small intestine by the HCl or what's known as the gastric acid, an enzyme protease and peptide to produce the amino acid. To use the amino acid, it must undergo the deamination process to remove the ammonia group. The carbon compound of the amino acid then will undergo the gluconeogenesis to get the glucose. The glucose will provide the energy for our body. Additionally, some amino acid can directly undergo oxidation and produce the energy. The ammonia produced from the elimination will be synthesized in the liver to produce urea and excreted out by the kidney.
Now, let's move on to tricarboxylic acid cycle. Starting our journey at pyruvate, amino acid like alanine, cysteine, glycine, serine, threonine, and tryptophan actively contribute to its formation. Next up is the alpha ketoglutarate phase. Here, arginine, glutamate, glutamine, histidine, and proline were found. Transitioning to succinyl-CoA, we find isoleucine, methionine, and valine. Moving along, we arrive at fumarate, where phenylalanine and triosine take center stage during glucose metabolism. Finally, aspartate and asparagine elegantly wrap up the metabolic dance, finding their roles in the oxaloacetate segment. This is the TCA cycle, and from this cycle, it will produce 2 molecules of carbon dioxide, 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of ATP. That's all from us. Thank you!